Now that we've looked at digestion, how the body breaks down larger carbohydrates into those smaller monosaccharide units, we'll look at how those units are absorbed and transported through the body. So remember when we're talking about the transport of carbohydrates, primarily we're talking about those three monosaccharides of glucose, galactose, and fructose. Of those three, glucose is the most abundant, and so it has the most significant implications, but all are important. So each of these molecules can't flow freely across the cell membrane. So in this picture on the screen here, you see the cell membrane right up on top. So up here is the cell membrane that we're referring to, those the outside of the cell. So this would be where the nutrients are coming in from, and then we're trying to get those monosaccharides across into inside of the cell. So when we look at this, basically there's two mechanisms that can be used, either active transport or facilitated diffusion. So in this figure, the Part A is showing active transport, and B and C are two types of facilitated diffusion. So remember, when we talk about active transport, that means that it requires ATP. So there's an energy requirement for active transport, whereas facilitated transport doesn't require ATP, it doesn't require energy, but it does require a transporter. So if we start by looking at active transport, so this can be used for, so we're just talking about this part right here on the left for that active transport. That can be used for both glucose and or galactose. And one unique aspect here is that, excuse me, sorry, erase some of this mess I have. One unique aspect there is that sodium is also required in this active transport mechanism to transport glucose and galactose across the cell membrane. So essentially there's two binding sites, one for sodium and one for glucose or galactose, and the sodium has to be bound in order for the glucose or galactose to be transported in. So this gives kind of one idea of where there's multiple compounds necessary for absorption to happen. One area you might think about this in is, for example, in a sports drink beverage that would have sodium in it, which is certainly to replace electrolytes, but also thinking about that's also going to ensure adequate sodium is there and binding so that the glucose, the sugars in the sports drink actually are effectively transported across the membrane. So that's one mechanism, is active transport. The other mechanism is facilitated transport, which involves glucose transporters. So in here, in this image in part B, they're looking at GLUT2 as the glucose transporter. So there's a family of transporters for glucose that are referred to as GLUTs, G-L-U-T. And each of those, we'll look at them a little bit more detail, but each of them are specific for particular substrates and are more active in certain cells in the body. So here, looking at GLUT2, transporting glucose and or galactose could be used for either, and the GLUT5, which is specific to fructose. So a little bit different transport mechanism or transport carrier for the fructose. And we'll again look at that in a little more detail in just a moment. This image represents a very general structure of glucose transporters, or GLUTs, and I just want to point this out just kind of to highlight a few, again, general aspects. This isn't specific to one particular transporter. So these little pieces here are that cell membrane, and remember we have those, the little tails going into the middle, so it separates out the hydrophobic and the hydrophilic aspects of the cell membrane. And so the transporters, they actually span throughout the membrane. So this is the whole transport molecule actually goes kind of in and out of the membrane. So it can be responsive to differences on the outside of the cell. So for example, if there's a high glucose concentration, or if there's a sense of medications, those types of things that can sort of interact with that external environment. And then it spans to the inside of the cell. Of course, it needs to be kind of on both places so that it allows those molecules to go through the cell membrane and get from the outside to the inside, which is the whole intent of the transporter. So this is just kind of a refresher or information on sort of how those transporters look in the cell membrane. Your book has a nice section on the various glucose transporters, and kind of discusses how there's a wide range of them that have been identified. Some of them we know more about and seem to be more predominant, or at least we're more aware of their predominance. So the things I would like you to kind of be familiar with from this is that there are different glucose transporter proteins, and each of these are specific to a particular substrate or multiple substrates. So in here, and we just talked about this, tells what substrate it will bind. And we just talked about, for example, that GLUT5 um, uh, transports fructose but not glucose. And then the other thing that's important to recognize is that there's different major sites of expression. This doesn't necessarily mean that it's not expressed anywhere else in the body, but these are the major sites that we're aware of expression. So for example, GLUT1 you're going to see more in the nervous system and active in the nervous system, where GLUT4 is really known more for its activity in muscle tissue and adipose tissue, although it is also present in some other tissues like cardiac tissue, as well as some white blood cells. So this isn't that they're only active here, but really the major site of expression. But being aware that there are differences in what each of those transporters bind and where they are predominantly active. GLUT4 tends to be particularly interesting because it is the insulin-dependent glucose transporter. And so essentially, often we think of insulin as transporting glucose from the blood into the cell, but the reality is that insulin just triggers the mechanism that allows then the GLUT4 transporter to move to the cell membrane and actually transport the glucose in. So both insulin and GLUT4 are necessary. Insulin is what kind of triggers the reaction, but GLUT4 is really what does the transport. So again, there's a nice description in your book on the GLUT4. Good to be familiar with how that functions to some extent. Here's an image again of looking at GLUT4 in a little more detail. So just to kind of highlight a little bit of how it works, essentially GLUT4 is stored in what's called glucose storage vesicles, or GSVs. So each of these little circles is representing a GSV, or glucose storage vesicle. And those are produced within the cell, stored within the cell. So, so GLUT4 is always residing in the cell to some extent. And then when there's some stimulus, whether that's a presence of insulin or it could actually be exercise, also stimulates GLUT4 translocation and perhaps increased production of GLUT4, then these GLUT4 storage vesicles actually move up to the cell membrane where they dock and they fuse. So then they are in the cell membrane. So that's kind of what this is showing here. And we kind of just looked at the larger image of how those GLUT transport proteins span the cell membrane. So this is kind of a simplistic look, but it does go across the cell membrane. It fuses, which then allows that glucose to come through that glucose transporter, get into the cell, and then once it's no longer needed, so there's not a higher insulin concentration outside of the cell, or the exercise has stopped, that sort of thing. Basically, we don't need to get more glucose into the cell. Then those the GLUT4 transport protein goes back into the storage vesicle, and endocytosis happens, which is essentially bringing it back into the cell so it can be stored for later use. So this is specific, shows specifically a little bit how GLUT4 works. But I think one thing that's important for us to remember is that this process is triggered by insulin. It's not actually insulin that does the transporting, it's the glucose transporter that does the transporting of glucose into the cell.